On behalf of the organizers, I have great pleasure in welcoming you to this morning session. We have three presentations dealing with the scientific and also the basic linguistic and evolutionary point of view. The first speaker of the day is Professor Shanmugasundaram, whom all of you know, and he is going to give us a general idea about the science of genomics. Professor Shanmugasundaram.
The light in the Good morning to all of you. I was asked to do the job of presenting genomics to a mixed gathering. So I am not going to go into the details of genomics, but I am trying to say what genomics really mean to a common man. Next slide, please. We all started with a field called as genetics. What is genetics? Genetics is the science of heredity. So this is a long history, and this deals with the science how like begets like, how the son is looking like the mother, or how the daughter inherits the characters from the father. The next slide. What is heredity? Inheritance of characters from one generation to the another. I am giving most of these kind of definitions from Webster's dictionary because I don't want to go into the kind of definitions given by varied academicians, because that may perhaps end up in confusion unless they explain under what context they were using such definitions. Next slide. What is a gene? One of the biological factors determining heredity. So, because that also has undergone a lot of variations in terms of defining a gene, what we used to do 10 years before, and at the advent of gene therapy, how it's all going to change. But for a common man, this is one of the biological factors determining heredity. Next slide. Why don't you put on the mic? I can talk without. Genome. Genome is said to be the nucleic acid, whether it is DNA or RNA, of an organism or a virus. So what is a genome is the basic footprint of total information. So that is the informatics of a biological system, and that's whenever it is expressed, we see its manifestation. We see its manifestation in terms of several characters. So genome is a fundamental information that is present in an organism, and till date we don't know any organism that exists without an information of a genome. Next slide. Again, the genetic material that contains all the genes. If we accept genes as a fundamental unit of heredity, and they are responsible to give rise to various molecules which are eventually involved in giving a particular character to an organism. And what is a genome is nothing but kind of a pearl necklace. The pearl necklace, if you take it as a genome, each individual pearl is equivalent to a gene. But only difference here, it's not homogeneous pearls, it is all different. So each pearl is a different, and that's the basic minimal difference in the genome. Next slide. The genetic material that contains all the genes. Now when we define genome, what is the basic difference when people were talking genetics, and what is the difference that we are talking genomes today? People who are trying to understand the master molecule that was present in every living cell, which we believe contains the entire information of how the cells should divide, to grow and function, that they were able to understand the genome even without looking at it directly. So it was an indirect observation. So genetics, when the field started, they were trying to understand the function of the master molecules, not looking at it directly, but its ability to express and the characters that is reflected. That was the basis on which people understood the genome without directly looking at it. But then it is not just, genome is not a material that contains only genes. They contain genes, they also contain other information within the genome. As much as a pearl necklace, it has pearls and also in between string. So it is a combination of both. So genome is nothing but the master molecule that contains information which is expressed, which could be understood by people who are interested to study the genome through its expression. And also there are regions in the genome which perhaps express, which we cannot understand by its character outside. So genome is a mixture of both. Next slide. What is meant by genomics? Because the omics is a word which goes with a lot of prefix, like people started using in the last 2-3 years. They call us genomics, they call us proteomics, they call us peptidomics. So all these things are come into play. At least taking genomics as a word, what does it really means? The study of genomes as a whole. So study of genomes as a whole is coming under the topic genomics. Next slide. When we talk about genomics, what is called as a structural genomics? That means we try to understand how exactly the macromolecule or the information molecule or the master molecule is made up of. And how do we know that? There are very sophisticated techniques used to sequence the entire genome. So structural genomics is a field of study where the developed countries, having attained a greater facility of infrastructure, and also they of late automated many of these facilities, and in an automation they were able to sequence entire genomes. So structural genomics is nothing but sequencing the entire genome and getting that information, and also now all these genomic informations are available in the public domain. Next slide. What is functional genomics? The study of parts of genomes that function in a given organism or in a cell at a given condition and at a given time. So functional genomics could be perhaps referred back to the field of genetics. So genetics was perhaps the part of the functional genomics. So whenever parts of the genome express, that expression is reflected in characters which could be observed, which could be analyzed, and which could be understood. Next slide. How do computers function? It's a very basic fundamental question using just two quantities, 0 and 1. What do we mean by this? in the recent years, people have created an independent language, which we call it as the computer language. And to create a computer language, they used only two functions, 0 and 1. Either it should be with a 0 or it should be 1. So, by alternating 0, 1, just two variables, they were able to create many languages, computer languages, and all of us are aware that even supercomputers were created using only two functions, and they were able to perform mega events and mega functions. Okay, with this kind of a background, the next slide, how do biological systems function? If you can appreciate the kind of achievements that was made by the computer scientists in the past, now looking at the biological systems, they use 4 quantities. So if 2 quantities or variables could be used to generate a language, and that could perform all kinds of function, including precisely determining the time of a satellite starting and landing. Everything happens precisely. So you can now understand the way in which biological systems have evolved using 4 quantities, and the repetition of this variables has created unbelievable manifestation, starting from simple viruses, from bacteria, fungi, lower plants, lower animals, higher animals, and we even to the human. It's only 4 variable system. Next slide. Why one should sequence genomes? So what do we mean by sequencing the genome? What one does, these 4 variables, whatever I have mentioned in the previous slide like ATGC, how exactly they are present? It could be AAA and then a T. What does it mean? Or it is AT and C. What does that mean? Or it is CCC. So only these 4 functions keep on repeat varying, and that has given a message and an information, and the biological systems add an ability to decode that information and perform a very specific function. So the biological systems do possess an ability to decode the information whatever they have and then function. So sequencing of the genomes means to understand the information directly. To give you a kind of an example, if there is a paragraph of information presented in a particular language, and somebody tells the information, you can still understand it. But when you look at the paragraph yourself and then dissecting how many words, sentences it has, and how many words in each sentence, and what are the letters that constituted the word, that means getting a total information of the number of letters involved and the number of letters aggregation to form a word, and the number of words which formed a sentence, and couple of sentences which formed a paragraph, which gives us a piece of information. But segregating any one of them is not going to make any sense, but putting them in a particular order, in a particular sequence, in a particular way, it conveys a particular information, and that again would vary depending upon the time and space, because the same kind of information will not give us the same kind of an understanding. That would vary versus time and space. So why to sequence total genomes? Because we want to understand all the details as it is present in a living organism is the purpose for sequencing the entire genome, and that has been done all over the world in spite of the fact it's an expensive process. And today what we have, many genomes of microorganisms were totally sequenced. And perhaps to sequence a small genome of a bacterium would cost something like, say, 10 to 20 crores. But then 40 to 50 of these bacterial genomes were sequenced. We do have couple of animal genomes totally sequenced, plant genomes sequenced, and human genome is also sequenced. So this has happened in the recent past. Next slide. What does genome sequencing mean to us today? So this all happened, and living in a developing country, we have to look at the entire kind of an information and a scenario and what exactly this kind of an experiment mean to us. It's a huge business opportunity. If somebody asked me a simple question, people wanted to sequence genome, maybe they want to understand how genomes, the information is present, yes. And then what is the academic relevance? Well, we can use it for academic purpose. But that is not the intention with which the genome sequencing projects were funded by the developed countries. And who invested a lot of money in genome sequencing is not even the government sector. It is not just the academic sector. Of course, the academicians of different universities and the national laboratories were involved in sequencing genomes of organisms, but who funded this particular exercise, which is a large investment, is the industries. Now, why do they do that? Sequencing genome is not a simple academic exercise. It's going to be the biggest business opportunity. Genes were patented in isolation. Now that's the real kind of an issue that the people in a developing country should really understand and appreciate. Whenever genes are isolated, it's possible to isolate genes. There are simple basic techniques. It is also possible to sequence individual genes which are responsible for given characters. And whenever they thought that particular character function has a commercial value, immediately they legalized their entire research. Once upon a time we used to think research is meant to generate new information and new knowledge, and it is meant for the society, and then they should use it. True. But now comes the commercial aspect of the entire issue. Those countries who sequenced the genes first, they claim they have the priority right, and they would legalize it and say whoever wants to use this particular gene for a particular information has to pay a royalty. So genes were patented in isolation. Now what is happening today? Genomes are being patented. So the issue is, even developing countries could not do anything. We never had a mechanism to interfere with the developed countries and tell them, you just can't do that. But then we never had. We signed all the kind of documents and then accepted in principle whoever sequences a gene in whatever part of the world, and if he files a patent, that is applicable globally. And now genomes are being patented. So what does genome sequencing mean as today is a very big political concept. It's not just an academic concept. I'm sure all the discussions are going to be for today and tomorrow is going to be to discuss the basic academic issues concerned with the origin of a particular race, or whether you go through languages or the origin of the place or whatever. But what is I am trying to present it to you is the real angle with which the developed countries are interested is not to understand any of these issues. It's all marginal. They encourage developing countries to discuss it. You discuss all the marginal issues, keep doing it and be happy. Now what is their interest? We keep legalize the whole thing and say to the entire world, we have done it first and we have an authority over it, so you have to pay for it. Next slide. Those who sequence a genome first, They have a right to use the genome information for commercial ventures. The developed countries know that the developing countries would shout. So they are very clever what they have done. Whatever information they have, they have thrown it in the public domain. So they said, look, you are all countries where you can't afford to do this kind of research. Fine, it's very expensive. So developing countries are supposed to worry about catering the basic needs of the society and people. So we understand your problem. So we have done it. But after all, the information is free for all. So they throw the entire information in the public domain. It's available in the internet, so people can use specific websites. You can access the information at free of cost. But you can only access the information. You can use it for academic purpose. You can enjoy looking at the information. You can compare informations of two organisms and then propose new concepts and do academic work, but you cannot use it commercially. Now that's the basic issue. If any country wants to use an available genome information for a commercial purpose, they have to pay a licensing fee to that group of scientists or industry or the country which claims that they were responsible to sequence the genome of a particular organism. Next slide. Why do we worry? What is the commercial value here? I'm constantly saying there is a commercial value if we know about the genome. What's the commercial value here? Gene or genes and the products would be the most expensive commodity in the global market. I think all the audience are present here. What does this mean? What is going to be the most expensive thing in 2005 or in 2010? It's not a metal. I think all of you will appreciate whenever people want to establish in their family and life, they say buy gold, keep buying gold, because gold is any time you can use it back. It's always better than the paper which you call it as currency. Keep metal. And those who can understand platinum is better than gold, they started buying platinum. Okay, a metal. Or people said the best thing that you can invest your money is buying stones. Buy diamond, buy a green stone or a blue stone, which is the most expensive. 10,000 rupees a stone, 20,000 rupees. Somebody say yes, a stone for 1 lakh or 100,000 or 200,000. So people thought, what is the most expensive thing you can buy and possess? Is a metal or a stone. But that is all, peanut figures what we are talking. What is the price of a gene? I'll give you an example. Take the gene that would give us a product called as insulin. So insulin is a protein. And the gene, there is a gene which makes insulin in an animal cell or in a microbial cell. And to give you what really insulin means to us today, how much money Government of India is spending per year to import this protein called as insulin to serve the society is 1,000 crores. I think all of you should appreciate we are investing 1,000 crores of rupees every year to import insulin. Next question is, are we not producing insulin? No. We are not producing insulin in India. We are only importing it. Next question, why? We have talents. We have brilliant people. We have people who are brilliant biologists. We have people who are brilliant molecular biologists. We have brilliant people who are biochemists. We know what is business. We know what is industry. Why is that we are not producing insulin? After all, it's a small thing. You isolate a gene and then why don't you start an industry and produce insulin? We have not done it for the simple reason, legally we can't do it because the first company which cloned the insulin gene from an animal and a human, they have a patent right. So, even though we can do it today, at least for 11 to 17 years, there is a patent protection. For that reason, insulin could not be produced in India, and this country imports insulin from those industries who hold the patent, and then they produce this protein and distribute to all over the world, and the net profit of an insulin producing company is $1 billion. So it's a $1 billion business. So coming back to what is the commercial value here? If only one is intelligent enough, shrewd enough, and understand the economics of science and the economics of the molecular biology, the economics of the genes per se, one gene and one of its product could fetch you an income of $1 billion. So that's the commercial value of genes, and patenting them and getting their products in the industry and marketing all over the world, and that creates a fortune for countries who hold more number of patents. So we are not going to have in future wars where we need weapons. That time has gone. It's an intellectual war. It's a war on intelligence. If you are intelligent, you know where to invest your money, and if you know how to isolate the genes as on a priority basis and make global patents, that would decide, it looks, the economy of the countries. So I want you people to appreciate what we are discussing today is not just an academic topic. Of course, it is an important, interesting academic topic for biologists and molecular biologists. But it's a topic for a country, it's for the political system, it's a topic where it's a huge money spinning biotechnology event. Next slide. Well, I want to pose a question to this audience. Having said what it really means to isolate genes, or what it really means understanding the genome and patenting it, now comes a real question which has to be answered by the academicians and elite of a developing country. To whom does genome information belong? That's a very basic, fundamental question. Does it belong to an ethnic group that perpetuated it? Because for a number of thousands of years, in a particular place, by a particular society, the biological information was maintained and sustained and perpetuated. So, to whom it belongs? Or does it belong to an ethnic group that understood it first? That's a basic fundamental question. So, information is available, and there is always a group sitting in a developed country. They say, we are the first who understood, 1 minute sir, we are the first who understood the information, so it belongs to us. So, this is a question for the audience and also the speakers who are going to participate for today, tomorrow, and after tomorrow, and then arrive at a kind of a strategy and arrive at a kind of a recommendation that has to be clearly given to our politician and statesman, and then raise an argument. Okay, the information is available in the public domain, but then you say that we are not capable of using it. Next slide. So what is the future? to reach an agreement between nations to use genome information for commercial purposes. That has to be agreed upon, otherwise we will end up paying huge cost for these kind of molecular products. Is the last slide? Next. Okay, anyway I'll show only one and then just click on one. So this is just to show, when we talk about genomes, I have represented just only one chromosome which has got two arms, one on the top and you have an arm. This is called as one arm of the chromosome, this is called as the other arm of the chromosome. What I am showing is, whenever this actually gets inverted, we always talk about inversion that happens. That is, the information is getting inverted within one part of the chromosome, and that would result in a varied function and involve in mutations. I had something like 20 animations anyway. We will have time in the evening to say it. So with this, ladies and gentlemen, I hope I made myself